afternoon. My name is Aparna Naik. I'm a rising senior biology major at St. Bonaventure University. And this summer I've been working in the Altindus lab, and my project is focused on characterizing the insulin signaling properties of double-chain viral influence. So recently there has been some research on the concept of viral mimicry. So it is known that viruses invade host cells to reproduce, insert their genetic material, and hijack cell machinery. And they're very sneaky because they use many different strategies to avoid being killed by their hosts. And one of these mechanisms is viral mimicry, which is a concept that some viruses create certain peptides known as viral insulin-like peptides, or VILPs, as I'll address them in my talk, that mimic human hormones such as insulin. So my PI, Dr. Emma Altindus, actually found that 4 VILPs from the Iridoviridae virus family actually activate human insulin receptor and stimulate insulin downstream signaling. So we tested single-chain VILPs from this specific virus family. So because insulin is heavily involved in diabetes, our lab is focused on determining the role that these VILPs could potentially play in the development of this disease. So diabetes is a huge worldwide issue as it affects about 422 million people in the world. In America, 1.25 million people have type 1 diabetes, and this number is expected to grow to about 5 million in the next 30 years. There are 2 types. Type 1 is an autoimmune disorder, which means that the body essentially attacks itself and its own beta cells in the pancreas, so insulin cannot be made. But type 2, on the other hand, is when the body still produces insulin but does not respond to it as well. So even though insulin is still present in the body, the insulin receptors are not being activated as often. Insulin and IGF-1, or insulin growth factor 1, are structurally similar peptide hormones that are involved in the regulation of cell growth and metabolism. And the difference between them is that insulin is double chain and IGF-1 is single chain. So what does insulin do in our body? When a person's blood glucose levels are very high, the beta cells in the pancreas will release insulin into the blood and lower blood glucose levels. It performs this function by stimulating glucose uptake in fat and muscle cells. So for my project this summer, the purpose was to characterize insulin signaling properties of double-chain viral insulins. And the overall goal is really to understand the function of these double-chain VILPs and why do certain viruses create these molecules in the first place. And hopefully with that understanding, we can elucidate the role that these VILPs could play in the development of diabetes. So for the first half of the summer, I performed cell culture on mouse preadipocytes, which are fat cells. And these cells overexpress either human insulin receptor, IR, or insulin growth factor 1 receptor, or IGF-1R. Then I proceeded to perform dose response and time course insulin signaling experiments. And for dose response experiments, I added different concentrations of either insulin or IGF-1 to cells in a 6-well plate. As you can see here, 1, 10, or 100 nanomolar. And I would repeat the same procedure for the time course experiments, but at different time points, 1, 2, 5, 15, and 60 minutes. And prepare the protein samples of those cells, and using a BCA protein assay, test the protein concentration of our samples, and finally prepare the samples for gel electrophoresis. So for the second half of my summer, it was really dedicated to performing Western blots. And the first step is to perform a gel electrophoresis by loading the protein samples into the wells and transferring that gel to a membrane, then blocking the membrane with 5% non-fat dry milk. And the purpose of this step is to remove any non-specific antibodies that could bind to the membrane. And then overnight, we incubated the membrane with 3 primary antibodies. The next day, incubating with secondary antibodies, then adding the substrate, and finally developing the membrane using autoradiography film in the darkroom. And to test if we had any proteins, we would perform a Ponsel stain on the membrane and Kamasi blue stain on the gel. And the purpose of a Western blot is to detect any proteins in your sample. So we were looking specifically for 3 important proteins in the insulin signaling pathway: AKT, ERK, and IR. So we first had to establish or optimize the conditions for Western blot. And after a few unsuccessful Western blots, we realized we had to start working backwards and start troubleshooting. So because we saw that the marker was visible on the membrane, we realized that the transfer step of the gel to the membrane was successful. So then we proceeded to change another variable, which was a solution in which we incubated our antibodies. So we first used 5% non-fat dry milk, but upon recommendation from our colleagues, we switched to 1-XPBS, or phosphate buffered saline, but we did not see any bands after that change. So then we moved on to thinking that it was a problem with our secondary antibody and changed the brand. And we did receive a very strong signal, and because of that result, we decreased our exposure time with the film from about 10 minutes to 1 minute. And finally, we received this result. Even though it's not that pretty, it really helped us to gear us in the right direction. So at the top, you can see dark bands at around 250, and those represent total insulin receptor. And the faint bands highlighted in red represent the beta subunit of the insulin receptor, which is what we were looking for. But this result really helped us to realize that the proteins in our samples were not being reduced properly. Because if they were reduced, we would see a band for AKT at around 60 kilodaltons and ERK at around 42 kilodaltons. So after that result, we performed a Kamasi blue stain on the gel, and as you can see in the right corner, the protein stayed on the top of the gel, which again confirmed our theory that the proteins in our samples were not being reduced properly. So DTT and mercaptoethanol are important reducing agents that are supposed to be in the sample buffer, and they help maintain protein activity. But when we checked our sample buffer, they were not included, and it was the reason that our proteins were not being reduced and why we were not seeing clear, distinct bands for the proteins at their respective molecular weights. And this is an image of what you should see when you perform a Kamasi blue stain on a gel. So after we added DTT to our samples, we finally received these beautiful Western blot results. So on the right, you have an image of phosphorylated proteins, and you see a nice dose response at 10 and 100 nanomolar for insulin and IGF-1, which is what we expected, and also for AKT and ERK. So since we added different concentrations of insulin and IGF-1, 0.11, 10, and 100, we expected to see them only at 10 and 100, or the higher concentrations, because the small concentrations, 0.1 and 1, are too small to be so too small for insulin or IGF-1 to activate insulin receptor and IGF-1 receptor. And on the right side, you see total proteins, which served as our loading control. And as we expected, they are they have the same thickness throughout the gel. So what did I learn throughout this process? Well, number one was that one of our secondary antibodies did not work. And number two is that the proteins were not reduced in our samples due to the lack of reducing agents in the sample buffer. And the overall goal of our project is really to understand the function of double-chain VILPs and why do certain viruses create these molecules in the first place. And with all that information, gain a better understanding of what type of role these VILPs could play in the development of this disease, and also hopefully create better, faster acting insulins that could help people with diabetes one day. And because most of my time this summer was spent on troubleshooting and optimizing conditions for the Western blot, I was not able to fulfill the original aim of my project, which was to test the double-chain VILPs. So with more time, I would love to explore that. And the overall goal is really to test the function of these double-chain VILPs. I'd like to thank everyone in the Altindis lab, my mentors, Dr. Altindis, Martina, Chin, especially Yasmin for being an amazing lab partner, the BCRU program, and the NSF for providing me with this amazing opportunity, my family and friends for supporting me, and everyone here for being a wonderful audience. Thank you.
Any questions?
Could you explain why there are two bands for Earth?
This slide?
Yes.
So there are actually two bands because there are two different isoforms or forms of the Earth protein. And so one is at 44 kilodaltons and one is at 42. So they both should show up, and we did see that.
Also on this slide, I noticed that you focus on phosphorylated proteins. Is there a reason for this?
So usually in insulin signaling experiments, to detect insulin signaling, you have to detect phosphorylation because the insulin signaling pathway is a phosphorylation cascade, which means that one protein phosphorylates another. So theoretically, if you see the presence of phosphorylated protein, that means insulin is activating that protein and that the signaling is occurring.
Thank you.
Okay.
Yeah.
Sure.
Or else you can't